Thank you very much for the invitation to come here to Christina and Robin. And thank you for joining your lunch with us here. We'll be talking about our first studies that we have done at the new center, plus a little bit of some data that we have recorded previously. So here you can see a highlight of Glasgow and beautiful sunny Glasgow, and we are hoping to host the OHBM meeting 2022. We are shortlisted amongst two others, and so cross the fingers. I think the decision will be done this week. So apart from the beautiful conference center that we have here, we also have now the ICE center that you already mentioned. This was the original plan, and actually you can see that this picture I snapshot last week before I left to show you two things. First of all, it's there. The ICE center is up and running, and also it's very sunny. It can be very sunny in Glasgow. So inside we have the new 70 Terra, and so it was number 4, and we now have since last month now a upgrade already. So it is full CE certificate. So you see it has a new cover. It's a 32-channel setup. And I'm going to highlight one equipment that was developed in Glasgow by Shajan, and that is a new head coil that is, instead of having, so the advantage of the scanner is that it has a single channel mode and a parallel transmit mode in which it can be run for more demanding tasks, but you need to switch between two different coils. This is a coil that you can use for both setups. Shajan Benamayu has developed this. He was originally in Tübingen and joined us 3 years ago. And it's an amazing coil. It's parallel transmit and 8-channel transmit and 32 receive coil. And you can see here the first image done last last month, just before ISMRM meeting. And some of the features is it's not so tight as the other coils, so it has some more space, which is good for us for visual stimulation. And in comparison, now you can see that to a commercial coil, it has just a much better coverage by having these parallel transmit coils that can also be used in the two different modes. And you can see beautifully how the coverage, for example, of the cerebellum is at 0.3 millimeter. So what are we using high-resolution MRI for? Well, in my lab, it's all about fMRI and especially the cortical feedback. And to investigate cortical feedback to, for example, primary visual cortex, which we have been focusing on for quite a while now, is interesting site to look at because it receives the cortical feedback from relatively distributed areas of the cortex, which is very typical. Any area in the cortex has a bit of a chance of 66% estimated by Kennedy to receive input from any other area. So there's a high interconnection, and we want to understand what the cortical feedback is doing to a sensory area like primary visual cortex. So we conceptualize V1 as being very high in the hierarchy because it's, of course, in the feed-forward stream very low, but then it receives all these expert areas feeding back to it. So if they have something to say, it might be very useful and interesting. So in the lab of Henry Kennedy, they have investigated and compared the kind of streams in 30 years of literature in monkeys, and the kind of main message is if you look at the layers, you have a main feed-forward route that sticks to layer 4, layer 5, so the mid layers, layer 3B, they are forward connected. And then there are two feedback streams. One is in the very upper layer, the superficial layer, where there are no cell bodies, but lots of synapses that are receiving remote input. And there are layer 2, 3 in the upper layers, and in the deep layers 5, 6. So those are the blue areas. So there's the hope with increasing high-resolution fMRI to 0.8 millimeter isovoxel, for example, or below millimeter voxel resolution, that we can kind of tune into the different messages. And now it's kind of an empirical question how good that works. So I'll show you one of our first study. This data is from Minneapolis, and it triggered this kind of line of research in collaboration with Federico De Martino, Rainer Goebel, and Isa Yacoub, and many others. Just highlighting, we do always retinotopic mapping. We map out this lower right visual field quadrant in this case, and also the boundary area in blue. Then we sample from these patches in V3, V2, and V1 at 6 different layers, and we assign each voxel to one layer. This is a bit rough estimate. It's not always, it's just an estimate. And then we have different families of voxels of the different depth layers, and we train them with support vector machine and MVPA analysis to tell us if they have some information about the visual input. So there's a trivial case in which there's actual feed-forward input. I highlight this kind of population receptive field of the neurons of the voxels that we are looking at. And so it's trivial for a support vector machine trained on 3 runs to predict the 4th run. But here's the non-trivial case in which we mask the feed-forward input, and the only way in which information could come to this area that is informative for the visual scene is through cortical feedback and long-range lateral interaction. And what we can see is that the feed-forward information is at a ceiling level 90%, and it does, so there's lots of information. But for the feedback situation, most of the layers shown in the different color codes are now not getting any information, so the feed-forward information is stopped. It's always white, just the white doesn't give information. But in the most outer, in the superficial layers, we have significant information feedback. Now, one question I've always been asked is that trivial because bold EPI signal is of higher amplitude in the outer layers? And here's the comparison average over 4 subjects, that the amplitude and the bold response indeed is bigger in the outer layers, but the relation between amplitude and MVPA classification is non-trivial. So here you see the increase, whereas the information plateaus, and here you see a very slight increase, whereas a defined increase in the MVPA. But I want to look into this a little bit more. So here's the information, MVPA information profiles, as I call them, and here you see the bold activation. So it's actually the only one subject that has this increase, because it's basically a baseline experiment. Here it's always white, so there's no on and off in those voxels that are looking at this region. And therefore, the information is quite distinct to the average activation profile. Another line of argument that we did in Minneapolis, we used a spin echo sequence, GRACE 3D, which doesn't have the feature of increasing neither in the occluded nor in the really functional feed-forward stimulated situation. It's flat, higher activity, and the feed-forward plateaus, whereas the feedback is only in the outer layers. So the different arguments to say that actually we are getting some different distinct information. We have escalated this whole line of research now in various ways. Andrew Morgan, who has also a poster here, 872, he will talk about related data. We have scanned subjects for 10 hours, exposing them to 192 full images and 192 occluded images, and now we have a much better idea of the kind of information that's in the top layers. And we replicated this original finding. You see the rainbow colors again. The very top layer, the red one, has most of the information that is related to cortical feedback. And we have modeled this kind of feedback and thought about how we can explain that best. And we compared it to behavior of line drawings. And so here you see line drawings. And those are the best way of conceptualizing what the kind of activity pattern that we can find in V1. So to cut a long story short, the paper is coming up. It's at bioRxiv already. This is about a related study only using 24 images, and this one is still in the process. But it shows that the mind is kind of making a prediction. If you occlude something, And this effect is called amodal completion. You don't have a visual illusion. You don't see the rest of this image coming through. But when asked to draw, to make a drawing, it's very precise and very consistent across subjects. And we have also escalated that within the Human Brain Project. We are collaborating with 2 animal labs that are doing rodent imaging. And we link this, we collaborate with Matthew Larkham, who has linked this to a theory of layer 5 pyramidal cells that have the property of receiving 2 integration zones, one the apical dendrites and one more the proximal dendrites. And they have an integration principle that combines the potential feedback information and the feed-forward information in this super additive way. This is from Patchclamp. He has also done in vivo recordings in somatosensory. So tickling a rat that expects to be tickled will activate these superficial layer dendrites. They can be inactivated by optical imaging, and then this effect doesn't change the activation pattern. So really, but what this really shows is that there's a very strong cortical feedback receiving those apical dendrites in the superficial layers that makes a crucial difference. Christian Liefeld is recording for exactly the same occlusion paradigm that we have that I've just presented now in rodents to be informative for actual models. Then we can model what's the source of the fMRI signal in comparison to this rodent data, where you are able to see those individual tufts and apical dendrites. They are marked here with calcium imaging during those conditions in which we have feed-forward information or just contextual feedback information. But these are longer stories. What I want to show you now today is the first data that we have recorded, the full study that was recorded now at the new scanner in Glasgow. And here we focus on 2 internal processed mechanisms. One is a visual illusion, the neon color spreading that you can see here beautifully. You see this red and green square on top of those rings. And Johanna Bergmann, who's here, she came to my lab to start the study comparing visual illusion to something that is even more internally generated with this mental imagery. So we asked the subjects just to imagine a red circle or a green circle, and it was absolutely dark in the scanner, so it's just the pure instruction to imagine this. And we wanted to compare mental imagery and the illusion. Both are internally generated, and if we find evidence in V1, it's sure that it is through cortical feedback. It's nice to see where this feedback is coming from. So to map out, we have this illusion. Those are our regions of interest. That's where we want to see the signal. We mapped out again in V1 those areas in 16 subjects. And now we do MVPA analysis again. We train on 3 runs to predict the 4th run, and the performance is shown here. And what we can show, the performance, the task is for the classifier to predict which color of the illusion was present, green or red. And you can see that only in the superficial layers, this prediction is above threshold. So illusion, again, like in our previous study where it was more like filling in of a scene, is you can pick it up in the superficial layers. Now, a first control condition is to outline an object, and that takes away some of this illusion of the square. Now you think it's maybe behind, or it's at least less than originally, by keeping all other conditions very similar, classifying between this green square or red square behind this object. Now kills this kind of feedback in the upper layers. There's maybe still something, but it's not significant anymore. Another way of controlling is now to take out, to rotate these inducers. This gives no rise to any green or red objects in the middle. And indeed, classifiers are incapable of predicting this. So now comes our imagery task. No, first a perception task, so just as a baseline and comparison. So we actually show red and green disks, and classifier performance is close to ceiling again. Now comes the imagery task. So we instruct subjects to imagine in the center, and we give them some indication of the size to really imagine red or green disks. And at this location that we have looked at, again, it's very flat. So MVPA classifier cannot say which color has been imagined. But then we swap the analysis to the center part. So if I can go back and forth, so our region of interest was around the contour, but if we go now, if we now go to the center part and ask the same question, what we can see here is the MVPA classifier is significant in the deep layer, in the deepest layer. So it has a different profile now in the 16 subjects, and but doesn't classify for any of the other control conditions. So it's constrained when it's constrained the analysis to the center, it's focused to the imagery, but not to the other conditions. So this is a summary. Imagery in the center can be picked up in the deep layers, whereas the modal completion or neon color spreading can be seen in the outer layers. Two ways of comparing our results. There are two studies from the lab of Peter Rosamer with depth electrodes in monkeys. And here's the version this lab has been using to create a figure ground edge. So you can see here the figure that's shown to the monkeys. And in this figure, the lines are oriented in a different direction to the background. And this creates an enhancement of the response after a certain delay time of 100 milliseconds that is then sustained. And if you look at the layer profile, they were able to record at different depth layers. You can see this peak in the superficial layers that correlates with this increase of activity around the line. They don't find this further into the object. It's more around the line, and this fits perfectly to our results. And the second result, the memory or the mental imagery, they have a version in which it's hard to instruct the monkeys to do mental imagery. I mean, this is something very easy for our subjects. Although I have to say, Johanna did some extra trick to map out with binocular rivalry those subjects that are able to image very precisely, and we pre-selected them because they have then a prime. They are primed towards a green or red stimulus in this binocular rivalry. It's a beautiful way of sorting subjects that are good at mental imagery. So here the monkeys had a tracing task. They were trained to follow the line and then do a saccade to the end point. And in this version of this experiment, the line disappeared. The monkey has to wait. There's a mask in between. Then it appears again, and it has to reconstruct with a mental image how they are connected. And so you see then the response in depth layers recording that there's a certain peak in the deep layers. We don't see the superficial one. There are differences in various tasks. There's also lateral interaction, maybe in this one more than in ours. But so there's some relation to it. And from this, I want to just give a call or highlight one more study in the lab from Angus Payton. He's also presenting a poster on Tuesday. We train our subjects in a virtual reality room to maneuver between the different rooms. We then present movies of those trajectories. They're moving out of one room, but they have seen a cue before that was triggering an expectation, because they before the experiment starts, they hear turn left or they see turn right. And then while they're moving out of the one room, they already anticipate the room they are going to. Now, this is very common. I think, when we move around in our house, you go to the kitchen. If it looks like your bathroom, you would be very surprised. You have this internal already anticipations carrying around all the time. So in order to read out whether you have this anticipation also in V1, we do again the occlusion, because otherwise there would be so much overflow of feed-forward information. And now in this occluded area, we ask the question, the room you're going to, is there any consistent information? Can we train a classifier on 3 runs to then brain read the 4th run? And actually, yes, we find evidence in area V2 that is able to anticipate this kind of prediction of the room you're going to. In 10 seconds later, it's a block design. And this whole effect goes away if you don't give the cue left or right. So we replicate all of this. Without the cue, you don't have this effect. And with a layer-specific MRI in V1, this is still ongoing, and you can see more of the poster. But again, it seems that we have more information in the deep layers. That's not too surprising because you have, again, some memory, some imagery, some prediction, and this might be a counter stream. So to wrap up, the Matthew Larcombe proposed in the TICS paper 2013 that these layer 5 pyramidal cells are playing a key role to integrate in visual perception, for example, color, motion aspects in recurrent loops that recurrently loop back to V1 and then integrate these kind of features. Conceptually, he argues that might be a very good way of looking into, supporting neuroscience to look into these layer specificity, maybe even more important than looking at single neurons, because these cell body obsessed neuroscience might have been misleading to some extent, because it's actually the integration that matters, and in fMRI it's the energy consumption that matters around the synapses. And we argue that it's important to make conceptual models, more of this kind, in which these layers feature very importantly, and you have different sub-modules that integrate information. This is one example. Here are further potential examples. And we argue in this paper, in Frontiers, in more detail, why this is an important conceptual novelty and why layer resolved neuroscience might be very, very important. And just to wrap up my talk, I've shown 2 things. One is there are counter streams in visual cortex. These counter streams contextualize information. They give rise to perceptual illusions like the neon color spreading, but they also give rise to predictions in occlusion paradigms where you're expecting a certain feature to be behind the occluder. And while all of this is going on, and 48% of the audience has been done something while listening to the talk, because that's the overall average. We do always mind wandering, we do other things. And so while you are predicting how to hold a cup and all of these perceptual tasks, you also think about other things. And these other things may also be related to anticipating the next room you're going to, use your memory to create mental imagery, and this is also happening in the same substructures. And so the big question is, how are they kept apart and how does it work together? So I hope to follow this line in future and update you more as we go along. Thanks to my lab for creating these wonderful, conducting these wonderful studies, and some of them are around, and please come to the posters to ask them more questions. Thanks.